Good morning, students. Good morning, everyone. Welcome to Biology for Neet aspirants. In this video tutorial, we are going to discuss about C4 cycle. In the last video, we have discussed about C3 cycle, light reaction and C3 cycle. In this video, we are going to discuss about C4 cycle. C4 cycle is otherwise called Hatch and Slack pathway because it was discovered by Hatch and Slack. The discovery of C4 cycle in monocots such as sugarcane, there are differences between C3 plants and C4 plants. The plants conducting C3 cycle are called C3 plants. The plants conducting C4 cycle are called C4 plants. The plants of C4 plants are monocots such as sugarcane, maize, and sorghum. These are C4 plants. Has indicated that these plants have solved the problem of photorespiration. Remember, Rubisco is an enzyme which involves the reaction both carboxylation and oxygenation reaction. Due to the oxygenation, there is a waste process called photorespiration occurs in the C3 cycle. C3 plants occurs in the C3 plants, whereas this wasteful process is very energy consuming process. This has to be put shut down to this photorespiration process. So C4 plants are evolved a new system of photosynthesis process, that is C4 cycle. We are going to discuss now. The carbon dioxide is fixed in the mesophyll cells. The initial product being A4 carbon compound, the process is called C4 pathway of carbon dioxide fixation. Here, the first formed stable carbon compound is C4 compound. C4 compound. That is oxaloacetic acid. Oxaloacetic acid is a C4 compound. It is a first stable compound found in C4 plants, whereas in C3 plants, first stable carbon containing compound is phosphoglyceric acid. Phosphoglyceric acid, it is a 3 carbon containing compound. It is a 4 carbon containing compound, hence it is called C4 cycle. Hence C3 cycle is C3 cycle. Photorespiration occurs in C3 plants. Photorespiration occurs in C3 plants, that is Calvin cycle. It is named after who discovered the cycle, that is Melvin Calvin. Whereas C4 cycle was discovered by Hatch and Slack. Calvin cycle, which leads to 25% loss of the fixed CO2. This fixed CO2 is loss percent is 25%. Among 100%, 25% of the fixed CO2 is lost due to the photorespiration. Photorespiration occurs in C3 plants only as the enzyme Rubisco catalyzes both carboxylation and oxygenation. It occurs because of the enzyme Rubisco. Rubisco catalyzes both carboxylation and oxygenation. It catalyzes the both reactions, carboxylation reaction and oxygenation reaction. The reactions of the initial acceptor of molecule, that is RUBP. RUBP is the initial acceptor. In C3 plants, RUBP is the initial acceptor. As both light reactions and carbon dark reactions occurs in the mesophyll cells of C3 plants, it becomes essential for enzyme Rubisco to catalyze both oxygenation and carboxylation reactions of RUBP simultaneously. Why in mesophyll cells, both carboxylation reaction, both light reaction and dark reaction, both light reaction and dark reaction occur simultaneously in mesophyll cells. So it has to undergo both carboxylation reaction and oxygenation reaction. So due to the oxygenation reaction, there is loss in fixed CO2 because of the presence of because of the presence of more oxygen concentration in C3 plants in some times, while compared to the carbon dioxide. When the carbon dioxide level is less than the oxygen concentration, then Rubisco undergoes wasteful process called C2 cycle, that is photorespiration. However, in category of C4 plants, nature has evolved a mechanism to avoid occurrence photorespiration. So, nature has evolved a mechanism to overcome this wasteful process, that is photorespiration. It has evolved another type of mechanism, that is C4 plants, C4 fixation, which is thought to be harmful process. Photorespiration is a harmful process. So through evolution, C4, while compared to the C3 plants, C4 plants are comparatively evolved plants. Two Australian botanists, Hatch and Slack, in 1966 discovered that there are two types of chloroplast in sugarcane. In sugarcane plant, that this is C4 plant, C4 plant, it's sugarcane plant. There are two types of chloroplast are present. One type restricted to the mesophyll cell and have normal grana. Mesophyll contains normal grana. These chloroplasts carry Hatch and Slack or C4 cycle. Hence, Hatch and Slack cycle or C4 cycle has been found in most monocots. Hatch and Slack cycle is mostly found in monocots and some of the dicots. The plants having C4 cycle are known as C4 plants. The plants having C3 cycle are called C3 plants. Most of the monocots are C4 containing plants, and some of the dicots may contain the process C4 cycle. There are two types of chloroplast are present, that is two types of chloroplast are present in sugarcane, restricted to mesophyll cell and bundle sheath cell. There are bundle sheath cells also present in the C4 plants. We will going to discuss now. C4 path requires the presence of two types of photosynthetic cells, that is mesophyll cell. It requires two types of cells, mesophyll cells and bundle sheath cells. It requires two types of cells. One is mesophyll cell and another one is bundle sheath cell. Bundle sheath cells are arranged in wreath-like manner, because wreath means garland. Wreath means garland. It are arranged like wreath-like manner. This kind of arrangement of cells is called Cranch anatomy. Cranch means wreath. Cranch means wreath. This type of arrangement is called Cranch anatomy. In Cranch anatomy, mesophyll cell and bundle sheath cells are connected by plasmodesmata. Both mesophyll cell and bundle sheath cell are connected by plasmodesmata. There are some connections. Cytoplasmic bridges called plasmodesmata are present. This is mesophyll cell, this is bundle sheath cell. These two are connected by plasmodesmata. In C4 plants contain dimorphic chloroplast. Two types of chloroplast are present. Chloroplast in mesophyll cells are granule chloroplast. Chloroplast of mesophyll cells are granule chloroplast, whereas in bundle sheath cells are agranule chloroplast. Why this type of division of labor occurs in C4 plants, we are going to discuss now. Granule chloroplast are present, agranule chloroplast are present. In C4 plants, mesophyll cell and bundle sheath cells are present. In mesophyll cells, light reaction takes place, whereas in bundle sheath cell, C4 cycle takes place. That is, fixation of the carbon dioxide and production of the glucose takes place in the bundle sheath cell. In order to divide photorespiration, in order to stop the photorespiration process, Oxygen is impervious to oxygen gas impervious to bundle sheath cell. It is not pervious to oxygen and any of gases that are going to release during light reaction in mesophyll cell. These are released out by the stomata. Mesophyll cell, mesophyll cell in mesophyll cells, light reaction takes place. To to conduct the light reaction process, granule chloroplast are needed. Whereas in bundle sheath cell, a granule chloroplast are present. Here does not require any granule chloroplast to maintain the C4 cycle. The granule chloroplast contain thylakoids which are stacked to form grana as formed in C3 plants. However, in granule chloroplast of bundle sheath cells, grana are absent and thylakoids are present as stroma lamella. In mesophyll cell, granule chloroplast are present. They are arranged stack-like manner, whereas in bundle sheath cell, only stroma lamella are present. They are not arranged in grana or thylakoid. They are not arranged in stack-like manner. They are arranged as stroma. The presence of two types of cells, granule and agranule cells, allows the occurrence of light and dark reactions separately in each type. Light reaction occurs in mesophyll cell, whereas dark reaction occurs in the bundle sheath cell. In order to conduct like this, there is division of labor occurs in mesophyll cell and bundle sheath cell. Here, release of oxygen takes place in one type. Oxygen released in mesophyll cell, whereas fixation of CO2 fixation catalyzed by Rubisco enzyme occurs in another type of cell. In C4 plants, maize, sugarcane, etc., light reaction takes place in mesophyll cell. Light reaction takes place in mesophyll cells, whereas carbon dioxide assimilation takes place in bundle sheath cell. Carbon dioxide assimilation takes place in bundle sheath cell. Such arrangement of cells does not allow oxygen released in mesophyll cells to enter the bundle sheath cell. This type of arrangement does not allow the oxygen to pass to the bundle sheath cell. So, because bundle sheath cells are impervious, they are not pervious. They are impervious to gases. They does not allow any oxygen into bundle sheath cell. So wasteful process photorespiration can be stopped by this mechanism. So C4 plants are mostly evolved than C3 plants because of this mechanism. Now we are going to discuss step-by-step process of the C4 cycle. There are 4 steps involved in the C4 cycle. That is, first one is carbon dioxide. First one is carbon dioxide capture in mesophyll cell. First, we have to fix the carbon dioxide in mesophyll cell in C4 plants. The primary CO2 acceptor is phosphoenol pyruvate. The primary acceptor is phosphoenol pyruvate, a 3-carbon molecule present in mesophyll cell. It is a phosphoenol pyruvate is a 3-carbon compound present in mesophyll cell. The enzyme responsible for this is phosphoenol PEP carboxylase. The PEP carboxylase is enzyme responsible for the fixation of the CO2 with the help of PEP. Importantly, mesophyll cells lack enzyme Rubisco but contain PEP case. PEP case is a phosphoenol pyruvate carboxylase which has high affinity towards oxygen. This PEP case has high affinity towards oxygen. It has more actively towards carbon dioxide than oxygen. Whereas Rubisco enzyme performs, whether carbon dioxide is more, it performs C3 cycle, whereas oxygen is more, then it performs C2 cycle, that is photorespiration. Whereas in PEP case, it has high affinity towards carbon dioxide. It does not conduct the process, that is C2 cycle. The product of the first step is oxaloacetic acid. First formed stable product is oxaloacetic acid. It is a 4 carbon containing compound. The first stable compound C4 hence is known as C4 pathway. Hence it named as C4 pathway. It is also named as hash and slag pathway by the hash and slag pathway who discovered this process, hash and slag. So first carbon dioxide acceptor. First carbon dioxide acceptor in C4 plant. Carbon dioxide acceptor in C4 plant is PEP. Carbon dioxide acceptor in C3 plants is RUBP. RUBP. It is 5 carbon compound. It is 3 carbon compound. Next, enzyme involved. Enzyme involved is PEP case. PEP case. Here Rubisco. Next, first stable compound. First stable compound is oxaloacetic acid. Here phosphoglyceric acid. 3-phosphoglyceric acid. 3-phosphoglyceric acid is the first stable compound. It is a 4 carbon containing compound. 4 carbon containing compound. This is a 3 carbon containing compound. These are the differences of C4 and C3. The stable compound of C4 plants is C4 pathway. Although plants take CO2 gas inside the CO2 rapidly combines with water to form bicarbonate ion. This CO2 combines with the bicarbonate ion HCO3 minus ion. CO2 combines with H2O to form HCO3 and O3 minus and H plus ion. PEP carboxylase uses this bicarbonate ion as an actual substrate and catalyzes the addition of HCO3 to PEP. This PEP combines with HCO3 minus ion in presence of the enzyme PEP carboxylase to form oxaloacetic acid, that is 4 carbon containing compound. Phosphoenol pyruvate, 3 carbon containing compound, combines with HCO3 minus in presence of the PEP carboxylase to form oxaloacetic acid in presence of 4 carbon containing compound, first stable compound. This step can be summarized by the following chemical equation. Phosphoenol pyruvate plus CO2 in presence of phosphoenol pyruvate carboxylase oxaloacetic acid is formed. In first step, that is carbon dioxide capture, that is oxaloacetic acid formation. This is carboxylation reaction. Formation of C4 acid. Oxaloacetic acid is quickly converted into other 4 carbon containing acids such as malic acid or aspartic acid, depending on the species. With the mesophyll cells, these C4 acids are chemically stable and capable of being transported efficiently to other cells. So, oxaloacetic acid, by using the light reaction products, that is NADPH, converts into the malate ion and NADP+. Why it occurs? Because to maintain the division of labor, to transfer the light reaction products to the bundle sheath cell, oxalo acid combines with NADH to form malate. This malate again releases H plus ions to NAD plus in bundle sheath cell to form NADP plus. Like this manner, NADP and NADPH are interchanged between malate due to the presence of the malate and formation of the malic acid or malate ion. This is reduction reaction. First is carboxylation reaction, This is reduction reaction. Transport and decarboxylation in bundle sheath cell. In C4 acids, malic acid or aspartic acid are transported from mesophyll cell into the bundle sheath cell. Transportation occurs and decarboxylation also occurs. Inside the bundle sheath cell, which are rich in rubisco but lack PEP. In bundle sheath cell, rubisco is present, PEP is not present in bundle sheath cell. These acids are decarboxylated, releasing CO2. These acids, malic acid is decarboxylated. This is decarboxylation reaction. Decarboxylation reaction. Decarboxylation reaction. Malic acid undergoes decarboxylation. CO2 is released and again converted to the pyruvic acid. Releasing CO2 and 3 carbon compounds such as pyruvic acid. This is key step to increase the local CO2 concentration near rubisco. Near rubisco, CO2 concentration increases by this step and can be represented as malic acid gives rise to pyruvic acid. This CO2 involved in carbon dioxide assimilation or glucose formation. Rubisco, here Calvin cycle occurs. Here C3 cycle occurs. Calvin cycle occurs. The last step is regeneration step, that is PEP and completion of this cycle. The three carbon compound pyruvic acid formed in the decarboxylation reaction migrates back to the bundle sheath cell to the mesophyll cell. It migrates back to bundle sheath cell to the mesophyll cell where it is converted back into the PEP. This regeneration of the PEP from pyruvic acid requires energy. This process requires energy. Here ATP is required. For 2 pyruvic acid molecules, 2 ATP are required. 2 ATP are required and phosphoenol pyruvate is formed. PEP is formed. AMP and inorganic phosphate is released. The final reaction ensures the cycle continues. This PEP again combines with another CO2 or HCO3 minus ion to continue the cycle. So what is the energy expenditure in C4 cycle? In C4 cycle, 3 ATP is used in Calvin cycle and another 2 ATP is used in regeneration process. So total 3 plus 2 equal to 5 ATP are used and 2 NADH are used. 2 NADH are used for one CO2 molecule. For 1 CO2 molecule. For 1 CO2 molecule, 5 ATP and 2 NADH2 are involved, whereas in formation of the glucose, 5 into glucose contains 6 carbons. So 5 into 6, total 30 ATP are utilized and 2 into 6, total 12 NADPH are utilized. Total 12 NADPH are utilized here. So for fixation of the 1 carbon dioxide, 5 ATP and 2 NADPH are utilized. For fixation of the glucose molecule, 30 ATP and 12 NADPH are utilized here. This is the structure of the bundle sheath cell. This is arranged like garland manner, that is Cranch anatomy. This is the bundle sheath cell. This is the mesophyll cell. This is bundle sheath cell. This is vein. Internal structure of the vein. This is stomata. Photosynthetic cells of the C4 plant leaf. Transfer section of the C4 plant leaf anatomy. This is the flow chart of the C4 cycle. Once again we go through this cycle. This is mesophyll cell. This is mesophyll cell of chloroplast. This is bundle sheath cell of chloroplast. These two are connected by the plasmodesmata, that is some cytoplasmic bridges are present, that is plasmodesmata. Plasmodesmata. Initially, pyruvic acid, initially phosphoenol, this PEP, phosphoenol pyruvate, PEP, PEP combines with CO2 to form oxaloacetic acid. To form, this is a 3 carbon containing compound, and single carbon is combined to form 4 carbon containing compound. And then 4 carbon containing compound. This is carboxylation reaction. Carboxylation reaction. This oxaloacetic acid is converted into the malic acid in presence of the NADPH. In presence of this oxaloacetic acid is converted into the malic acid or aspartate in some plants. This malic acid is transported to the bundle sheath cell. Malic acid is transported to the bundle sheath cell. It is a NADPH is utilized here. NADPH is utilized here. It is a reduction process. Reduction process. This malate ion is transported to the bundle sheath cell. This malic acid releases one carbon dioxide. This carbon dioxide involves the C3 or Calvin cycle, C3 cycle or Calvin cycle, whereas formation of the glucose takes place in C3 cycle. In order to increase the carbon dioxide reaction more towards the bundle sheath cell, this mechanism is arranged like this. This malate releases carbon dioxide. This is the decarboxylation reaction. Decarboxylation reaction. So pyruvate is formed, pyruvic acid is formed or pyruvate is formed. This again transported to the mesophyll cell and cycle goes on here. This pyruvic acid is converted into the phosphoenol pyruvic acid. This pyruvic acid is converted into the phosphoenol pyruvic acid in presence of the ATP. Here again 2 ATP are utilized here. Total for fixation of the 1 CO2, 5 ATP and 2 NADPH are utilized. Okay, that's it for today. We'll going to discuss CAM cycle also in our next video. Thank you so much for watching this video. Please subscribe to my channel.